Thanks a lot for the opportunity to be here. So my name is Tim Liu. I'm the CEO and co-founder of the company Senti Biosciences. Senti is a company that's focused very much on building the next generation of cell and gene therapies, and it's powered by about 2 decades of research into the synthetic biology field. The vision of the company is to enable more intelligent medicines, ones that take advantage of the full information processing capability of biology, where we can leverage the fact that cells naturally sense and respond to their environment and use that for therapeutic purposes. So as a mission statement, we're about 50 people in South San Francisco, very much all focused on coming together, trying to build the world's best cell therapies, gene therapies, based on our ability to design what we call gene circuits. As a brief history, I'll just tell you a bit about where the company came from. This company actually grew out about 2 decades of academic research from a variety of labs. My own background is trained as an MD Ph.D. scientist, and I've been on faculty at MIT for the last 9 years. And so I've seen, as a field, we started in 2000 with the first genetic switch that someone built into a bacterial system, to today when we can build all sorts of computing circuits, sensors, et cetera, into human cells. So back in 2016, together with a team of scientific founders and advisors that I'll introduce in the next slide, we felt like this was the right time to take that technology out of the lab and try to solve some of the major problems we were seeing with the first generation of cell and gene therapies, which are certainly really exciting. So we pulled together a team of executives and investors to help drive forward this vision. Philip Lee, my co-founder, chief operating officer, deeply experienced in the manufacturing side of things, is responsible for taking our programs and actually making them, translating them towards the clinic from a manufacturing perspective. Kurt Herberts was here in the room, very actively driving forward our strategic and business development efforts. As you'll see a little bit later, we believe this technology is quite broadly applicable to any cell and gene therapy application where you're thinking about enhanced control or programmability. And Gary Lee, who comes from us about 13 years, previously at Sangamo, last the VP of cell therapy there. So a lot of experience taking cell therapy into the clinic. We've been fortunate to be supported by a group of biotech as well as technology investors, closed our Series A, about $49 million in February of 2018, and that's allowed us to grow as a company, to build up our underlying technology platform for designing these genetic circuits, but also to enable us to take some of our programs forward towards the clinic ourselves. I mentioned we work very closely together with some of the leading minds in the field of synthetic biology. The vision here is that, as I'll show you in the next slide after this, we want to be able to enable cells to sense, respond, and adapt to their environment in a variety of ways. And that means that we need to be able to build sensors, we need to be able to build what we call logic gates inside of cells that can actually take that information and make decisions. And we need to be able to then have those cells do the right thing from a biology perspective to have the right therapeutic effect. So we've pulled together an advisory group to really lead us in that effort. So Jim Collins, one of our scientific co-founders, was the guy who developed the first genetic switch back in 2000, has really guided us in terms of where we think the technology is going to go in the next 5-10 years. Wilson Wong, who's at Boston University, has been doing a lot of next generation immune cell engineering, CAR engineering, which we incorporate into our therapeutic portfolio. Martin Fusenegger at ETH Zurich's done probably the most work engineering cells that can sense and respond, and demonstrating a variety of disease models in vivo that you can cure disease and make closed-loop feedback type therapies that can sense, for example, how much glucose there is to make insulin or sense metabolic levels in the body. Mo Khalil and Chris Voigt are also world-leading synthetic biologists developing platforms that enable these genetic circuits and switches. And finally, on the right side here, we have a group of clinical advisors, including Larry Fong, who's at UCSF, an expert in immunotherapy, and Michael Andreeff and MD Anderson, who's been a pioneer in cell therapies as well, translating those towards the clinic. So just to give you a little bit of an insight into what I mean by the gene circuit, essentially what we're talking about are multi-gene components that you insert into a cell. So we're not talking about simply overexpressing a single protein, but having multiple genes inside of the cell that actually can coordinate much more sophisticated behaviors. So we really like to break up all of our therapeutic applications into this workflow. On the left side here, we think about what are the inputs that we're trying to detect. So in the context of a CAR application, it would be, maybe we want to detect 2 different antigens and be able to build a system that can actually respond to 2 antigens before triggering activity of a CAR T cell. In the context of an immunology application, we think about being able to build sensors that can detect, high levels of inflammation and engineer a cell that knows how much of an output to produce and respond. So to enable these types of a variety of different sensing to happen, we've been building synthetic receptors that can actually take advantage and sense what's going on in the extracellular world and actually transduce that information to the inside of the cell, as an example. In the middle here, this idea of a circuit, that just means we can engineer cells that can actually incorporate multiple pieces of information together. So for example, going back to that context of being able to sense and respond to multiple antigens, we've built CAR systems that can actually sense 2 different antigens and give you like an AND gate behavior. We'll only trigger T cell activity if antigen A and antigen B are present, or systems that are not gate-like. So you can build cells that say only kill target cells that express antigen A, but don't kill cells if they express antigen A and B together. So that gives us the ability to build much more discriminative cell therapies. And finally, on the output side, clearly anything we can genetically encode into a cell we can express. And one of the areas that we're quite excited about, especially in the context of oncology or immunology, is to be able to actually express multiple payloads from cells, to be able to hit multiple pathways that are involved in those disease processes. So it's not just solely focused on cell killing, but it's being able to modulate, for example, T cells, B cells, and NK cells as part of that process. So from an engineer's perspective, why this is challenging is once you start putting together multiple genes together, it's about how to coordinate that activity and make sure the interactions that you're programming into the cell actually work properly together and don't break the system. And so we have a team of not just biologists, but a team of biological engineers, computer scientists, et cetera, that help us design, build, and test these gene circuits in very high throughput. And so we can actually build much more reliable and powerful type systems. So here's just an example of where we think this is going, and I think we'll talk about this perhaps a little bit later. In oncology, we're very much interested in building cell therapies that address some of the major outstanding problems in solid and liquid tumors. So on the solid tumor side, very much interested in engineering cells that can actually home to the tumor microenvironment and then locally change that microenvironment in a combinatorial way. And so we leverage our genetic circuits to build systems that can actually localize and sense what the microenvironment is, and be able to make decisions in that information in that environment, and then respond by modulating the immune system. For liquid tumors, we very much believe that there are not a lot of good CD19 BCMA-like antigens that are out there. Likely for many diseases like AML or for solid tumors, we're going to need to be able to sense and respond to multiple antigens. So finally, I'll just briefly mention we've built up a set of genetic circuits, a bunch of toolkits in the company that allow us to build cells that can sense and respond. We have technologies that are rheostat in nature, where we can use a small molecule to turn up or turn down the activity of a cell. We've been very much actively building and testing a variety of combinatorial immune effectors, pairs and triples that can actually hit multiple aspects of the tumor immunity system. And then finally, I mentioned logic gating and programmable CAR systems. And we have internally decided to focus our internal pipeline to develop oncology assets. We want to bring those towards the clinic ourselves, but very much interested in areas outside of oncology where we think the technology has a lot of applicability, especially in immunology, regenerative medicine, et cetera. We've published many papers in this area and very open to collaborating with companies in that space. So with that, I'll leave it there, and I guess we'll sit down and have some questions later. I'll leave this slide up there just to sort of stimulate potentially questions or collaborations in your mind. Thank you very much.
Okay, is this on? Okay, so thank you, Tim. I fear we're going to scratch the surface here. Maybe you have quite a team. You have a fresh look at cell therapy. I think what most people know about cell therapy is that Kite sold for a lot of money, and so far sales are disappointing. So, in your sense, in your view, what is the low-hanging fruit to make this a more general approach, and what are some of the thoughts on what are the first things to try, given your bag of tricks?
Yeah, I think from our perspective, we want to try to enable a much broader range of indications to be addressed with cell therapies and gene therapies in the future as well. I think as you heard great from the last speaker that if you're simply relying on constitutive overexpression of a single target or only relying on being able to sense and respond to a single antigen like CD19 and BCMA, you're going to naturally be limited to disease indications where that's the case. So from our perspective, if you really want to try to broaden these types of therapies to solid tumors, to areas outside of oncology, the bar for being able to discriminate disease from non-disease or to be able to have safety regulation in the case of non-oncology applications really goes higher. And that automatically is going to necessitate, we believe, more sophisticated genetic engineering, and that's what Senti is very much focused on. So for us, we're trying to tackle low-hanging fruit in oncology, where we think multi-antigen targeting is going to be really important to give you that range of discrimination, to give you a high therapeutic effect, and also tackle the solid tumor problem.
Okay, so you talk in broad strokes about regulatory elements. There are only really a handful that have gotten very far. How hard is it to make new ones, and where are you? I understand you're private, so you don't have to tell us a lot of the details. But how hard is this? Because there's really just a handful of switches. Some of them are 20 years old. So just your thoughts on.
Yeah, so it's not. I think for us, the biggest challenge has always been how to put these pieces together. So when you take a protein, you try to combine it with a synthetic promoter or a microRNA element and actually get all that to work together. What often happens is if you just take a naive approach to this, you get sort of unknown or undesirable interactions between those components that can screw up the system. So for us, it's really about how to figure out how to put those pieces together. We are doing some innovation around, for example, we have our own synthetic receptor technology that's been now validated across, being able to detect things ranging from small molecules like nicotine all the way to be able to detect proteins like PSA that we think is quite robust, and we've built those and optimized those ourselves. But I think the big grand challenge for the field is how to put these things together rather than, how do you get the world's best transcription factor to be built. That's the vision we look at.
A lot of optimization.
That's right. Yeah.
I think it's intuitive on the safety side. You just have your cells look harder at target cells. On the efficacy side, are the pieces known? It seems like I think there's something like 3,000 PD-1 combination trials. So does your technology make these experiments easier, or just your thoughts?
Yeah, so from our perspective, it's about trying to widen that therapeutic window, because I think there are mechanisms that are been tested in humans where you can see elements of efficacy, but because of your inability to localize whatever payload to a specific area, or your inability to discriminate better between what's a cancer cell and not, that's what's limiting your ability to dose high enough to get that efficacy. I think that's an area that we think is low-hanging fruit for us, where if we can widen that therapeutic window because we can discriminate better or have local effects, we can take advantage of some of the existing know-how that's already out there and has been tested in humans as the first sort of foray into the clinic.
So localization is a little focus. I mean, we've written a lot that the signal's there, you just need to focus a little because you can't deal with the systemic tox. So.
That's right. Yeah, so there's a class of molecules that have been in people that have that profile.
I think people know a little bit about cell design labs, mostly because of the price tag. Is that your nearest competitor? Is this a busy field? Do you feel like you have enormous runway?
Yeah, so I think Cell Design Labs probably was our closest spiritual competitor. Know Wendell Lim really well, as a good colleague in the field, and I think that really showed the value potentially of that type of technology. I think so I would say if you're looking for a closest comparable, they're probably the ones that we would point to as well. I think we believe we have a nice runway ahead of us, given that we've pulled together technologies from some of the other top leading labs in the area, and really are trying to solve the problem of how to do design, build, and test efficiently. Our vision is that the more times you go through that process, the better you get at building these systems. And because the core challenge is how do you put multiple pieces together, it's not something that you can just come in naively and try to do it and have it work well the first time. And so there's an economy of scale and learning that you get over time with this.
Okay, and I think like a lot of new science, it's likely to be expensive. Do you view the first big news to be a partnership, or do you think you would go it alone, or just your thoughts?
So we're taking a hybrid model. I think very open to discuss with people who are, and we're also in discussions already around partnering. Because we can be efficient designing these systems across a wide range of cell types, indications, et cetera. It's not too much off our back to be able to engage in some of those partnerships. So we love to chat with people there. In terms of cost-wise, we are envisioning using allogeneic cells in many cases for some of the work that we're doing. And so I think that's a way in which we're trying to also address that particular problem.
And then just as if we were to label you with one term, do you think you're mostly a synthetic receptor company? Are you a switch company? Are you a promoter company?
Yeah, that's a great question. So we really think of ourselves as the gene circuit company. And that is meant to encompass the idea that we're not limited ourselves to a single receptor or a single switch. It's about how to put components together. It could be a switch, it could be a promoter, it could be a sensor, but how to put things together to achieve that TPP that you're looking for. And that's, I think, ultimately, if you look at this diagram right here, this is the central node of what we bring to the table in terms of designing cell therapies and gene therapies.
All right, so thank you very much. I've had hour conversations with Tim, and you feel the same way at the end, but hopefully we got you going. Thank you.